All right, good morning, everybody. Let's get stuff sorted out. Everybody hear me okay? I keep getting a message from Zoom saying that my audio isn't working.
Yeah, you're fine, sir. We can hear you.
Cool. Thank you. Check, check, check. Do that every class. Wouldn't that be fun? Doo-ba-doo-ba-doo. Here we go. Get the chat. All right, I'm in my office because I have lab today, which is cool because you get to see some of my stuffed toys. That's one. Not sure who that's supposed to be. Maybe me. And this is one of the cutest things ever. Isn't that amazing? My daughter made that for Christmas a couple of years ago because I love giraffes. Second favorite animal. Last Christmas, I got not an avocado. Oh, shit. One of those things that are like armor plated, have cute pointy ears. See him run over on the side of the road a bunch. My brain's just not working right. Anybody? Armadillo. There you go. That's it. I don't know why I kept thinking avocado, obviously. That has nothing to do with it. Anyway. Okay, so today we have kind of a bit more of a recap type deal. Some good questions came up in other class, so it kind of shows that some of this stuff is worth going over, and it just depends on individuals as to what you need essentially. Okay, so this PowerPoint's really all about going from structure of DNA, basic structure, where I will get into much more detail later, and all the way up to a bit of an overview of mutations and kind of natural selection, I guess, speciation, like very kind of briefly. Okay, so one kind of critical thing that you need to know that's really tied into an awful lot of stuff, right, is the structure of DNA. And it's no real surprise that our understanding of how things work at a molecular level really didn't take off until we figured that out as, community, right? And so there's a lot of reasons for that, but we'll get into those later, essentially. So one of the things that's really worth pointing out is, well, there's a bunch of stuff really. Berenice, can you hear me now? Sorry, I just saw your. Okay, awesome. Yeah, just saw your message. Sometimes if I didn't catch your message or if it got buried, just I think there's like a hand up thing or something, or just unmute your mic and say, Professor, I asked such and such, or whatever. Okay, so first off, each of these is called a nucleotide. Technically, they're a nucleotide when they're not incorporated into DNA, right? Once they're incorporated into DNA, we tend to call them bases, which is a little bit confusing sometimes, right? So each nucleotide consists of a phosphate group, negatively charged. You'll have to forgive my shorthand. A ribose sugar, and a nitrogenous base. Right, the phosphate and the sugar are the same for all nucleotides, all DNA nucleotides at least. DNA and RNA are different, right? Does anybody know the difference between the nucleotide level at least? Actually, there's two differences between DNA and RNA. Bit of recap, see if anybody remembers that from prior stuff. Yeah, that's a good one. Uracil. So what does uracil replace? What's its equivalent in DNA? Thymine. Yeah, that's it. I always remember because it's like a herb, like thyme, I guess. Right. So exactly, thymine. What's the other difference? Why is it ribonucleic acid and not deoxyribonucleic acid? Good point, Isaiah, but not correct. RNA can actually be double-stranded. It can bond base pair with itself. Not in the same way that DNA does, but it can form double-stranded structures. RNA is actually arguably way cooler a molecule, if molecules could be cool, than DNA is. What else? Clues down here at the bottom, it's what's attached to the sugar molecule. All right, put you out of your misery. So, there you go, no deoxy. So deoxy means it's without a oxygen, right? So DNA, deoxyribonucleic acid, which is what this is, has a hydroxyl group only at the 3' carbon. Right. RNA has 2. One on the 3' carbon, same as DNA, and on the 2' carbon, right? Which is one thing that makes RNA a little bit more kind of prone to degradation, a little bit. Also allows it to do other funky stuff like sticking to itself. Anyway, these sugars and phosphates kind of join together. They join together by phosphodiester bonds. So essentially this 3' OH or hydroxyl here is required for the condensation dehydration reaction, which produces water and a covalent bond, right? And so between this oxygen and this phosphate and this phosphate and this oxygen are what are called phosphodiester bonds, right? Very stable covalent bonds. These bases, these nitrogenous bases, are what are different, right? That's what contains the information, essentially. The phosphate sugar backbone is the structure. The bases in between are the information. One thing that's also worth pointing out too, these numbers, right? 5 and 3', that refers to the number of the carbon number, I guess, which something is attached to, right? And you count them from the oxygen. Get over there. Right, so you start the oxygen, the first carbon, the base is attached to, second carbon, nothing, hydrogen, that doesn't count. Third carbon, OH, fourth, and then the 5th carbon is what the phosphate group is attached to. Now, because, oops, there's only an OH group on the 3' end, that means you can only add new bases to the 3' end. Yes, Infinity, that's right. Phosphodiester bonds are covalent bonds. That's one reason why DNA is stable. Not the only one, there's others, but that's one reason. So DNA has direction, right? 5' to 3'. DNA is read by RNA polymerase in that direction. And it's synthesized in that direction too, right? So DNA has direction, RNA has direction, and because proteins are translated from RNA, proteins have direction too. All of this becomes really important later on when we talk about various mechanisms of replication and transcription. Okay. So again, you'll have to forgive me. Sometimes I might repeat stuff that I've already told you because I can't remember whether I told you or the other class. So I'll tell you again, just in case. DNA has topography, right? It has a kind of structure like the hill country. South Texas does not have topography, right? It's flat, 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 flat. Kind of like where I grew up, super flat. Anyway, DNA has a shape, and that's because it's not even twist. It has a major groove and then a minor groove. It's not like twisting two bits of wire or twist tie together, right? And that structure is actually what's recognized often by proteins, not just the sequence of these bases, but also the actual structure. I think it's the tyrosine inhibitor, tyrosine repressor, actually binds to the major and major groove, right? So it kind of comes in like so and slots into these larger spaces. We've got 4 bases. We've got complementary base pairing. We covered this last class, right? Good. So you can all remember how many bonds are between a guanine and a cytosine, right? Excellent. How many between an adenine and thymine? How many between an adenine and a uracil? Yes, exactly, right? Doesn't hang around for long, obviously, right? Because DNA, RNA hybrids are transient, but still it's the same principle. Okay, what else did I want to talk about this? Oh yeah, and also another big deal is that, and I keep repeating this because it's important, is that DNA is anti-parallel, right? It goes from top to bottom, or left and right, right to left, whichever, in and out, out and in. Doesn't matter which way you think about it in terms of orientation, right? The 2 strands run in opposite directions because DNA has direction. That's super important for DNA replication. And it has really kind of important consequences for transcription too, which we'll get to in a second, right? Because you can only make DNA and RNA in one direction. And yet the structure of DNA is bidirectional and they go in opposite directions. I think Jake's having way more fun than everybody else. Okay. Jake, pay attention. Good stuff. All right, whoever that is off the screen, go, bye. Speak to you later. Unless it's a particularly alluring individual, in which case, yeah, it'll still be bye, see you later. Genetics is more important. Okay, anyway. All right, and so really, this is all kind of tied together in the process of gene expression, right? So the DNA doesn't do anything. It is dynamic, right? It doesn't stay in one form, but it's not dynamic in the sense that it wanders around doing stuff. It stays in the nucleus and that's it. It's very much like a library, right? Libraries don't get up and wander around. They're just a store of information that stays put. So even though DNA contains the information, right, which ultimately determines the phenotype of the organism, along with, the environment as well, it doesn't do so directly. It has to do it by some other intermediates, right? And those are RNA and protein. Protein, generally speaking, is the thing that does stuff, but it's not the only thing. There are a lot of RNAs, actually more and more as we kind of get to understand these things better. Hang on. There we go. That's better. RNAs play a really big role. They're actually really, really interesting. You might kind of go, oh, it's on about RNAs again. At least it's better than talking about pandas. But RNAs are super, super interesting, and they do a lot of cool stuff. But generally speaking, when we talk about things that do stuff, we're talking about proteins, right? And so the production of those is kind of collectively referred to as gene expression, right? And oh, actually, that's a point. I need to upload this, change this. Ha. It's been bugging me for years, and I've just been kind of too distracted to change it. So this is what's called the sequence hypothesis of molecular biology, right? The whole thing being referred to as gene expression. So those two are equivalent, synonymous. I think that's what synonymous means. And so we're really talking about the fact that DNA gets turned into, well, it doesn't get turned into, gets copied into RNA, and that RNA, depending on what kind of RNA is, gets translated into protein, right? So many times you'll hear this referred to as central dogma. And some of you who've had me before may know I've got a little bit in my bonnet about the correct terminology. This isn't actually a central dogma. Central dogma, which was proposed by Watson and Crick, is it doesn't go backwards, right? You can't go from protein to RNA to DNA. That was the theory, at least. You can actually go, there are a few things that can go, well, either in the lab or in nature, from RNA to DNA. Actually, does anybody know what can go from RNA to DNA? Sarah's looking excited. Was a big scourge in the 80s and 90s. Viruses, yeah, what type of viruses? AIDS, yeah, well, HIV, actually, yeah, you're right. HIV is one of those, right? HIV, shingles, actually, is also a RNA virus as well. Herpes, that's cool, I didn't know that. What are they called collectively? Does anybody know? It's kind of how I think of, you know, like those mum jeans that come up to your waist, and that's right, retroviruses. Okay, and so they're able to reverse transcribe, right? So reverse this process and turn their RNA genome, because they're RNA viruses, into DNA, and that can actually get incorporated into the host genome, right? So that's one reason why it's very hard. I'm not sure about herpes. I don't know much about it, to be honest. Pardon me. But that's one reason why HIV can, and especially shingles, can lay dormant for a very long period of time. My nan, my dad's side, my dad's mum had shingles. I think, like, you know, whenever she got stressed, she would just erupt again, as it basically came out of hiding in her genome, which is pretty crazy. Anyway, so the central dogma isn't perfect, but it's really just a thought concept. Oh, no way. Yeah, it's a really, really weird virus. I don't really hear very much about it anymore. It seems to be kind of like a, back where I came from anyway, a virus that old people had a lot. Yeah, I don't know. Anyway, so this whole process being gene expression or the sequence hypothesis, and it really consists of transcription and translation. Transcription making an RNA intermediate, which is mRNA, that's the messenger, that has to get, at least in eukaryotes, from the nucleus to the cytoplasm, where it's going to be translated. But also makes a whole bunch of other what are called non-coding RNAs. Type that on out. So these two examples here, rRNA, so ribosomal RNA and transfer RNA are good examples of non-coding RNAs. There are lots of others. So many I don't actually know all of them. Well, not even like a small percentage of them maybe. And so the cool thing about ribosomal RNA is that RNAs can be both a store of information, basically like DNA, and they can be enzymes, like proteins. And those are called ribozymes. Right. And ribosomal RNA is an example of one of those. So our RNA basically catalyzes the formation of peptide bonds and also removes the amino acid from the tRNA, which carries it into the ribosome. Okay, again, stuff we'll cover in the future. So another cool tangent, because it's fun to talk about. So this duality of RNAs actually makes it a top candidate for the origin of life, right? So this is something that I teach, well, pretty much never, here at least, in a course called astrobiology, right? Which is super, super fun. If you want to put on, pester the department chair to get it scheduled, because it is a super awesome course. Basically covering the entire history of the Earth, extraterrestrial environments, aliens, or maybe not aliens, stuff like that. So that's basically the RNA world hypothesis. Life started, oops, if I can spell that right. Basically because RNA, they think, or at least the predecessor of what we now know as RNA, is because it is a store of heritable information, right? You have to have information that can be passed from one, generation to the next. Otherwise, essentially, you don't have life, right? It doesn't work otherwise. And something that could make itself, because it can be an enzyme as well. They haven't quite yet been able to recreate that in the lab, but they've got pretty close. I think they've made a, I don't know, 25 or 30 nucleotide RNA molecule, like synthetically, that can make something like 80% of itself. I don't know the rest of the details because that's kind of whoosh, way of my head. But it's kind of proof of concept type stuff. Not the only hypothesis as to the origin of life. There are other kind of ideas, but it's a pretty cool one. Anyway, kind of a little bit of a tangent there. So first part of that process is transcription, right? Taking the information in DNA and copying it into RNA. And transcript actually is a good word because it's one that you're all familiar with, right? Your transcript, pardon me, is a copy of your courses and we'll get you in a second, Rebecca, and the grades you get, right? And so RNA is also a transcript of the information in DNA. It's not the same, but it is a copy of that information. Oh, got a good question from someone on the chat box about the percentage of our genome that is actually encodes useful info. So that's actually an interesting point, Rebecca, because we don't actually know what is useful info, right? We have a fairly decent stab at it. What's probably better to say is that our genome, a human genome at least, is about, I don't know, depending on how you count it, 3 to 4% protein coding genes. right? So I'd actually have to, I'm just changing the chat thing so I can type this up. So I don't actually know the exact percentage, but it's around there. So, 2% seems a little low, but I haven't looked at it for a while, so that might be right. Obviously, it depends on which species you're looking at as well. So it was always thought that the rest was just junk, right? Didn't contain anything of note. That's actually not the complete picture. There are an awful lot, a huge part of our genome is big part of the rest is viruses, transposons, things called pseudogenes, which are kind of result of duplications, and then the genes kind of die, they get mutated and they don't function anymore, stuff like that. However, there's an unknown part that contains non-coding genes. Right, so these are the genes which you can't find them very easily, right? So one way in which we find genes, right? If you sequence an organism and you want to find where are all the genes, right? What genes does it have, right? What you're actually looking for computationally is a protein coding sequence, right? Because it's relatively, I guess, I mean, I've never actually done that kind of work, easy to find the start and the stop codons of a open reading frame, right? So you go, oh, there's a start codon, there's a stop codon. What's in between must be a gene, right? Now, non-coding genes don't have that. And so, get you in a second, Rebecca. So it's really hard to find them. You can only really find them experimentally. And so recently we've begun to do a better job of that, and we're finding more and more non-coding genes which do things we don't necessarily understand. And so I'd be very careful kind of painting too broad a brush as to what our genome contains, because we don't actually know for a big part, which is kind of crazy, right? We've had the whole genome sequence for what, 20 years nearly, but we don't actually know. And so this whole kind of idea of junk DNA is a little bit of an outmoded concept. A lot of it is, but some of it, and we don't know how much, is not. Also, another question about viruses attacking mitochondria in the nucleus. Don't really know. I'm not much of a virologist, to be honest. I do know that some viruses, like retroviruses, do incorporate themselves into the genome. Also, others kind of hijack the different parts of the cell machinery to be able to replicate themselves at high efficiency. So that might be part of that, but I'm not really an expert. Oh, how does the mRNA vaccine work? Well, basically, the mRNA vaccine, which is like the Pfizer, Moderna vaccines, it's a really good question. I'm going to got in my mind to incorporate some COVID stuff later on into the class. I just haven't thought of how to do that yet. Basically, what the mRNA vaccine is a short piece of mRNA encapsulated in lipids, right, which allow it to get into your cells, like the ones that are in your arm and stuff. And it encodes a short piece of, I'm pretty sure, the spike protein, right? And so, let's see if I can find that. Not soy protein. Ooh, lots of cool pictures. So the spike proteins are these cute little red bobbly bits on the outside. Those interact with certain cell surface proteins on our cells and allow the virus to stick and then get into the cells somehow. I don't actually know how. And essentially, this is what our immune system sees, right? We don't see the proteins inside. We don't see really the capsid proteins all that much, or the envelope proteins rather. We see the spike proteins a lot. That's what we kind of recognize. And so the vaccine basically gets encodes a bit of that protein, which is, or different areas of it, which are very successful at being recognized by our immune system, right? They're kind of usually on the surface of the protein. And it tricks our body into making that protein. And essentially what our cells do is they get that protein and they go, look, to the immune system. This is a little bit of a, shortening of how it works. You have to take an immunology to figure it out in depth. And it holds it up and goes, here, look, they're called antigen presenting cells. And your immune cells go, well, shit, that ain't good. And it goes around and it basically makes antibodies to it. They don't necessarily use the protein to poke into the cell wall. That's more what are they called? Bacteria viruses? Shit, I can't remember. The ones with like the injection thing. They quite often, what they actually, that's it, phages, bacteriophages. Most viruses, there you go, let's draw up on the wall. Right, I'm going to have to end this tangent a little bit because, we got stuff to cover. So really cute little flies, kind of kept them up there. Oh, all right, let's go to this part of the board then. So essentially you have a cell like so and a virus which kind of sticks, right? And so your cell has all kinds of proteins on the surface of it. That's right, Kaylin, it's always fun. And the spike proteins recognize and stick to those proteins. And what that then allows it to do is essentially merge its membrane with that of the cell. And then the RNA, which is in there, gets into the cell and essentially tells the cell to make more of the viruses. Yeah, it's very much like a little Velcro ball. And that's how most viruses work. Not all of them, but most of them. Like HIV, for example, recognizes a protein on T helper 4 cells, for example, like a protein called CCR5. But anyway, what the vaccine is doing is basically putting a little bit of the viral genome, the bit for the spike protein, into your body. And so you're making it as if you were kind of infected like a virus, but you're not. And then those proteins get presented to your immune system as if they're bits of a virus, right? Which in a sense they are. And so you develop an immune system to that, develop antibodies. So that if you do get infected by COVID, essentially you don't have the 3 to 5 day lag time, which takes your cells to be infected, present those proteins to your B cells and things like that, or T cells, and for your immune response to kind of, step up. And in that time, the virus can actually replicate sufficiently that your immune response can't get rid of it. And so the vaccine is really like getting a jump on the virus. It's getting those antibodies there so that if the virus comes in, they're immediately recognized and kind of glommed onto by your immune system and neutralized or absorbed. Yeah, viruses are really interesting. Got a bit of a bias against them because they're not technically living, I guess. But they are, they're pretty, I think one thing is what I like about a virus is that they have just the bare minimum and nothing more, right? They are very efficient things, right? And they're exceptionally good at what they do, right? Because that's the only way that they can survive, even though they're not living. I don't know. They're weird things to talk about, to be honest, once you get into it. All right, so kind of getting back on track a little bit, which is fine. We've got time. It's fun to talk about this stuff. Let's make sure I didn't miss anything. Oh. Yes, so essentially what we have going on is, in transcription at least, right, you have to start the process, right, which is initiated by proteins, pardon me, that bind to and recognize regions upstream of the gene, right? So they're responsible for turning the gene on, or transcription beginning. And then once it does, essentially what happens, and I really love this image. It's kind of one of my favorites to work with. What happens then is the DNA gets, let's kind of scooch that out a little bit, split, right? So the two strands get separated apart. So you have like a little bubble. And one of those strands is used to copy the DNA to make the RNA, right? This strand is not the one that contains the information. It is called the template strand. And the reason being is that you can only make RNA, just like DNA, by adding nucleotides at the 3' end. So in this diagram, the information that's being copied is the one on this strand, right? So the strand over on the right is the coding strand. And so this is something that we'll come back to too. The one on the left, the one that actually has the RNA transcript kind of stuck to it, is the one that's being used to copy the coding strand. Because as you see, the direction of the mRNA from 5' to 3' is the same as the direction of the coding strand. Right, so again, this little complex here is anti-parallel. The template strand goes down, the mRNA strand goes up. And so if you compare it, right, we've got a thymine here in our coding strand. We've got a uracil, we've got an adenine, we've got an adenine, thymine, uracil, and so on and so forth. Right, so this is a really elegant kind of image. I really like it because it really gets at what's going on. And that we've already covered. And so basically this mRNA sequence is probably best put here. It consists of words, right? So the mRNA obviously consists of a series of bases, right? And those are really the letters, right? And they are actually represented by letters too. So those letters are organized into words. And those are called codons. And those are 3-letter words. I should have put that earlier, never mind. So these 3-letter words are what are translated by the ribosome and the tRNAs and the ribosomal RNAs into an amino acid sequence. And the thing that does the translating, really, like the Rosetta Stone kind of deal, are these tRNAs, right? And these are, I think someone said, I think it was Isaiah earlier, that, you know, RNA can't form isn't double-stranded. There's actually a bunch of, I think that's a tRNA. Let's go back to here. So if you look at tRNA, there's a bunch of regions of tRNA which is actually double-stranded, forms these things called loops. right? And these loops are really important. Yeah. Yeah, once you get kind of down to it, it's chemistry at the end of the day. I hate to admit that, but that's really what drives an awful lot of what goes on at the biological level. But anyway, these loops, and this is really what tRNA looks like, this three-dimensional structure, basically has an amino acid attached at one end, and at the opposite hand has an anticodon. And so the anticodon, oops, basically recognizes the codon, right? And that's how the mRNA is translated, right? So RNA can take on all kinds of funky shapes. They really can. And they can be dynamic as well. So they can kind of move. The shape is important for, so this one is a ribozyme. So it has a catalytic pocket. Some of them are involved in controlling gene expression in different ways. Very many different ways, actually. They can do all kinds of funky stuff. And essentially, this is kind of a molecular equivalent of the Rosetta Stone. Oops, get that out of the way. And so this is the genetic code, which tells us, okay, what word equals what amino acid. And you can see there are way more words than there are amino acids. There's 64 codons and only 20 amino acids. And so this gives us a lot of redundancy, right? It also actually means that you can have mutations in coding regions that don't change the protein. Because if you change the last base on several codons, like if you go from UCU to UCG, for example, you still have serine. Right? This is called degeneracy or degenerate genetic code. Also, as just a heads up, here are 3 codons that do not code for amino acids. These are involved in stopping translation. Right? These are called stop codons or termination codons. Anybody got any questions so far? We will cover this in more depth later. This is really a kind of get everybody up to speed sort of deal. I do love my coffee. You know, believe it or not, I didn't drink coffee when I was at university. I have no idea how I managed. Just remembered something. Okay, so just to kind of wrap up, Red Bull. Yeah, I did drink a lot of Red Bull. Actually, Rebecca, that's funny. I keep wanting my wife to make me a t-shirt that says "Life's too short for bad coffee." So maybe I'll ask her to do that for Christmas. Not super fancy, but not you know, the stuff that you buy in those large plastic containers for use in restaurants and stuff. I was addicted for coffee for 2 days. Well, that technically isn't an addiction, Sarah. You know, if we're being pedantic about it. Flavored, it kind of depends. Texas Pecan is nice. That's actually one of my favorite from HEB. Anyway, just quickly to kind of wrap up. Come on, get in there. Obviously, DNA can change, right? Actually, it can change in many different ways. The main way that we kind of think about it, particularly in terms of inheritance, right, is a change in the sequence of DNA. It's not the only way you can change DNA and have it inherited, which we'll get to again later. And that's a whole different field called epigenetics, right? But by and large, mutation is the main source of novelty, right? And this is important to get, it's random. So what part of your genome a mutation will occur in, whether it's by DNA replication mutations or external insults, such as, UV radiation or living in California, stuff like that. It's random where those occur. It's random what bases get changed. Sometimes bases get changed, sometimes bits get deleted, sometimes bits get added, right? All kinds of different variations on that theme, but that's the main source of novelty, right? In terms of change, right? And by and large, and this kind of comes back to, I can't remember who it was that mentioned about how much of our genome codes for protein, right? Because we have this large chunk of our genome, which is kind of baggage, I guess, right? Ancestral baggage of viruses and transposons and stuff. Most of those mutations don't have any effect, right? We just accumulate them over time. Actually, it's one way in which you can measure time over large spans of evolution, called the genetic clock. But those that aren't harmless, neutral, is what we call them, typically they're going to be deleterious or bad for you, right? Because if a protein coding region is changed, or a regulatory region, or something that affects the function and production of that protein, typically that's not going to be good, right? Because in most cases you have those genes and produce those proteins for a reason, right? And if they don't work or they're not there, then you're not going to be able to do that thing. And occasionally you have some which enhance the fitness of that individual. Those are pretty rare, right? And so this is something that's always fun to think about. Say just by random chance, you get a mutation in your germline, right? If it's in your somatic tissue, like, this bit, not your sperm or your eggs, it dies with you. Doesn't matter what it does. Even if it kills you, it still dies with you. It doesn't get passed on to the next generation. But even if you did, right, say we're kind of, I'm not going to use myself as an example because that's kind of gross. But say someone had a mutation in their germline in San Antonio. That would be in one copy of their DNA, of which we have 2. And so that mutation would be present at 1 in 1.3 million, right? And so there has to be some really strong selection pressure or a lot of time over which that selection pressure can work before that mutation, that one mutation, expands in number, so you can actually see it and it has an effect on the population. So it's really cool to think about it in that way, because it really hammers home the time part. Oh, Kaylin, yeah, so if you have a mutation in your skin cells, right, it may affect you as an individual, but it won't get passed on to your offspring. And so when you die, that mutation to your genome dies with you, right? So even if that mutation made you like the most, I don't know, attractive person in the world, or like the brainiest person or whatever, doesn't matter, right? Well, I mean, it'll matter to you maybe, but in terms of inheritance, that stops just there. So that kind of gets to this handy dandy chart down here. And so that's really the, I'm getting a question about things that could apply to the British royal family. I'll get to that in a second. So the whole germline versus somatic. So somatic tissue is anything that does not get or cannot get passed on to the next generation, which is basically almost all of you, like all of you as an individual, not all of you as a class. only mutations which are present in your germline, so sperm for males, oocytes for women, only those mutations can potentially get passed on to the next generation. I had a question about inbreeding. Inbreeding doesn't create mutations, it concentrates them, right? So it basically results in a higher frequency of particular mutations in a population or a family tree, but also a higher likelihood of homozygous recessive genotypes, which will show the phenotype. Quick question about lab. No, you can do the lab acknowledgement really by the end of this week. I'd do it today, or I'd do it in your lab session, whatever session that is. So if you're an odd section, like an O1 or an O3, for my lab sections at least, that will be yesterday or today. If you're an O2 or an O4, that'll be next week. For the other lab professors, you need to refer to their schedule, because I don't actually, the registrar does all kinds of weird stuff every semester, which I don't understand. So I can't say for sure whether it's which way round they have theirs. You'd have to ask them. And no lab coats are needed for first lab 1, either this week or next week. You do need them for lab the following week. The lab safety acknowledgement is in the lab 1 course content folder. You'll see there's like a little assignment at the bottom where you can download the form. There's no due date. I'll end of the week, I would say. Basically, so I don't have to nag you, right? Anytime before the end of the week that you have your first lab in would be ideal. There's another question too, which I think I missed. Berenice, no, you need to fill out a lab safety acknowledgement for whichever lab section you're in. And I'm pretty sure both of the others have posted this. I sent it out maybe Monday night or Sunday night, I can't remember. But they should have had it posted up already. So it applies to everyone that's in the lab. You can do an electronic signature. So I think there's something called CAMI, like where you can put in your electronic signature. Doesn't matter doesn't matter how you do it, as long as it is signed and uploaded to Blackboard. Obviously, whichever way is easier for you, because that, means it's more likely to get done. Cool. I think that's it, everybody. I think I would suggest focusing on what we cover, Sarah. Generally speaking, I think a lot of the stuff, like some of the odd bits and pieces that we talked about today, are really good for context, but tend not to be the main focus of the exam. Sometimes there are things that we are going to be in the exam that we talk about in class, right, that aren't in the PowerPoints. But they're fairly rare. Bio 1 would help too, but that's also part of what we're doing at the moment, is just to kind of recap stuff, make sure everybody's on the same page. But yeah, if you have the time, then certainly that would help as well. Cool. All right, everybody, if you have any other questions, feel free to bring them along on Wednesday, or just send me an email as well. It would be just fine and dandy. Have a great day. That was a lot of fun. I need to go and do some more reading into COVID. And yeah, see you soon.